Welcome back. Our next talk will be by Jasper, and he will be talking about AlphaFold.
Yeah, good afternoon. I think this talk is going to be quite a bit different than the other talks that you've had so far, because it's going to involve quite a lot of biology. So the title of my talk is AlphaFold, a new age for protein folding. And if everything goes well, you should see my slides changing right now. So allow me to introduce myself first. I'm Jasper. I am a PhD in computer science. So I'm not a biologist whatsoever. I just happen to have been introduced into the field, and now I'm kind of stuck there. So I did my PhD on deep learning for biological sequence analysis. And currently I'm doing a postdoc at the Flemish Institute for Biotechnology and the University of Ghent. And I was contacted by Kenneth. I've actually worked together with Kenneth a bit already about AlphaFold. So he asked me to do a presentation here on AlphaFold because apparently it's one of the most frequent software requests that he ever had. And I suppose quite a lot of you will have the request as well a couple of times already. So it's quite a hot topic in the field. So what I'm going to do here is try and give you some background about what this software does, why is it important. I'm also going to talk about what proteins are and what the software actually does. How did it come to be, and how is its use on HPC systems. So I'm first going to talk a bit about proteins and what are proteins. All of you know that we all have our own DNA. We are all very unique. And all those pieces of our DNA are actually codes to encode some kind of traits about our body. So our appearance, our phenotypical traits, as they say, like eye color or curly hair or whatever, is encoded in our DNA. Everything there is to know about how our muscles should contract, how our immune responses should work, which is quite important these days, you know. Food digestion is included in there. So really, our DNA encodes for everything. And it's not just from DNA that this comes, but it's our proteins that actually do the work. So allow me to talk about how we go from the DNA that's in our bodies to the actual functionality. So there's a couple of steps. And this is called the central dogma of molecular biology. This is a simplification because there is backtracking and everything in the system as well. But the main rule set is as follows. That's that DNA is transcribed into RNA. RNA is translated into proteins, and those proteins will then have some function in our body, in our cells or outside of our cells. Doesn't matter. DNA is made up of two strands of nucleotides. After transcription, this is RNA, where we have a single strand of nucleotides, and this is translated into a chain of amino acids. And one long chain of amino acids is what a protein consists of. Of course, it is not just code. There are many regulatory factors where certain things are happening in our body, what the circumstances are, other factors come into play. And I'm going to talk a bit about one extra factor that is the protein sequences. And that is that when we look at a protein, this is not merely a sequence like it is often put to paper, but a protein inside our cells, it actually folds up to a certain molecular structure. And it is that structure more than the actual code that makes it do a certain function in our body. It is going to bind to other 3D molecules, so it's important that it fits on top of them, etc. So that's one of the main takeaway messages here in this introduction. Like the code is not the most important thing. It is actually how that code folds up that is very important, and that is what AlphaFold is going to help us with. So the prediction of those protein structures has for the past 50 years or something been regarded as one of the holy grails of molecular biology. And that's because we actually have a lot of sequence data. Sequencing experiments are a lot cheaper and a lot faster than getting the protein structures. And if I put it visually, you can see that, well, in the most common, in the main database online, the protein database, PDB, there is some 50,000 unique structures when we filter for redundancy in those structures. And as you can see, we have a lot more sequences at hand. And it's the amount of sequences is also growing a lot faster than the amount of structures. So why do we have only 50,000 structures? It's quite a lot already, but there is a deficit. And that's because it's quite hard to achieve those structures. You can see a picture here of the main experimental setup with which those structures are calculated, and that is X-ray crystallography. And this is the machine that does it. As you can see, it's not just, it's quite an expensive piece of equipment. We're talking about $1 million equipment here. So those are not available in all labs. And it's also a labor intensive and quite a slow task. So when you really want to test a lot of proteins for their structures, we're looking for other ways of doing so. So 3D structure prediction, where can this aid us? Like at the most easy things, I'd like to propose the most easy tasks that come to mind when I think about 3D structure prediction, and that's that you can actually just generate a lot of 3D structures given a good predictor, low effort and very time efficient. So what you can do is, if you have a lot of drug candidates that you would normally screen for structures with those crystallography machines, what you can do is just before that you do that, screen and rank your candidates based on the structures that come out of an in silico computer predictor. This can save you a lot of money, a lot of time, and you still will end up with very good candidates. And they also help when you want to design proteins from scratch, which is one of the next big things in molecular biotech, given the rise of artificial intelligence in the last decade as well. That's that you can really design your proteins to do what you want, to make them producible and everything. And 3D structure prediction will also be a big aid on that front. So how has 3D structure been predicted until very recently? The basic principle behind this is that 3D structure is more conserved than sequence. So all proteins in our body are a result of millions, hundreds of millions of years of evolution. So the proteins that are in our body that take care of food digestion, let's say, are very much alike proteins that are present in a chicken's body, let's say, especially the structure, because those need to interact with certain pieces of food and whatever. Those structures are very well preserved, more so than the sequences themselves. The sequences can mutate without having an actual effect on the structure. So what's typically been done is homology modeling. And the principle here is that given a query protein sequence, like the one over here, you look in your database, in your structure database, okay, do I have something that resembles this sequence? So when you find one like that, like the one here in green and orange, what you can typically do is take the structure that goes with that, make a copy of it, and then fine-tune it based on certain physical modeling and statistical constraints, so that you end up with a structure very much like the other one that might fit the sequence of interest here. But of course, we don't always have those structures available. And the most important competition where those structure predictors are evaluated is the 2-yearly critical assessment of structure prediction, the CASP competition, which is where AlphaFold was first launched into the media. What do I want to say about this? 2 things here to introduce this slide. So I have here the results of the last 14 years minus 2 competitions at the end of this CASP competition. And I'm showing results here for free modeling accuracy. So I talked about those in my previous slide here, about those homology models, where you can, for a big part, make copies from another protein that we know already. Free modeling is actually an evaluation on targets where we do not have similar structures available. So this is a much harder task than just any target. And they're evaluated on a metric which is called the global distance test. And you can see this as follows. You have a ground truth protein, and what you do is you align your predicted protein to be similarly aligned as a ground truth, and then you see what percentage of my amino acids in this protein are within a certain distance. As you can see, a 93% GDT gives quite a nice match in the structure already. But as you can see, on average, the results for this free modeling accuracy were not very satisfactory so far. And that is where AlphaFold came in to help. AlphaFold is a beautiful piece of software which was developed by DeepMind. DeepMind is a subsidiary, an AI company, a subsidiary company of Alphabet. It was acquired by Google in 2014, when it was still Google, not Alphabet yet, for about $500 million. And they were in the news a couple of times already. In 2016, there was AlphaGo, which also has a YouTube documentary about it, which is very nice. I can really advise you to have a look at that. And that's where they used artificial intelligence to beat a 17 times world champion in the game of Go. So Go is a game with a much bigger board than, for instance, chess. And this makes it a lot more difficult to calculate, to make a computer get to play Go, because the number of possibilities that you would calculate for all the outcomes of the games is just ridiculously high as compared to, for instance, chess, where you can do that. And that's why we were able to beat chess at the end of the 90s, I believe, already. And beating the world champion of Go was only foreseen in like a decade from now. So they had some very nice breakthroughs there. They released AlphaZero, which was a lot better than Go, and which was a self-learning kind of algorithm. In 2018, at the end of the year, AlphaFold version 1 was released on the CASP13 competition. They kept on playing games as well. The Agent 57 was able to self-learn and beat human level performance on the Atari 2600 games. And then the event for what I'm going to talk about is AlphaFold 2.0 with a victory at CASP 14. So back to the results that I showed earlier. In 2018, AlphaFold version 1 was released, and in 2020, AlphaFold version 2 was released. And as you can see, all of a sudden, there is a lot more that can be done. We're here on all targets, so including the homology structures we already know, similar targets. There's a median of 92.4, and there's even a median of 87.0 in the free modeling accuracy. And you can assume that by the time you reach a 90% accuracy, we can talk more or less about experimental quality of structures. So this was a giant breakthrough, and the reception was also quite enthusiastic. So I have a couple of very important people in the field here. The most important one in protein structure prediction is probably Mohammed Al-Quraishi, who said, "I think it's over, one of the most significant scientific results of my lifetime." More people talked about the once in a generation advances. Some people talked about the problem being solved. Even the chess players, here we have Grandmaster Kasparov, who is happy that Google DeepMind is going to leave the chess players alone for a while. And 2 weeks ago, Science released the protein structure prediction, not only AlphaFold, because there were a couple of other predictors as well, as the 2021 breakthrough of the year. So the impact to the field is expected to be ridiculously big in the next couple of years. So let's talk about AlphaFold itself a bit more, because we go from a sequence to a structure, but a lot of stuff happens in between. And the basic principle on which AlphaFold trains itself is coevolution. So you just, as a user, give one sequence in input, and what AlphaFold is going to do is it's going to look in similar sequences. Okay, do I find any coevolutionary patterns? A coevolutionary pattern you can see like this. If you have a couple of sequences that are very similar, but that always change certain positions at the same time. As you can see from the colors in this example, this is often the case because they're in contact. And if one of those contacts changes, the other also has to change because the structure should be maintained. And that is one of the basic features that AlphaFold bases itself on. So the first step in the whole AlphaFold process is, given an input sequence, it's going to do a multiple sequence alignments. So this is done by a search in a genetic database, which is installed on the machine where AlphaFold is run. And it's going to just look for all sequences that are somewhat similar to the input sequence. It's going to align them so that you know which position change and which are just randomly changed, which have those coevolutionary patterns, etc. And those are done later, going to be given to the deep learning modules of AlphaFold itself. Another thing that is done is that the structural templates, so those homology models, are also fetched in a structured database search. And those templates are used as a starting point, but the researchers at DeepMind say that as soon as your multiple sequence alignment is actually deep enough, so you have enough sequences, 30 plus, they claim, those templates are less important. So after we have those multiple sequence alignments, they are run through a couple of different trained AlphaFold models. So there are 5 AlphaFold models for which the parameters are released currently. And those are just completely the same architecture, but trained with different randomized initializations. And then at the end, once the structures are predicted, there are some extra constraints and physical fine-tuning that is done using a software package called Amber. And one very cool thing about the outputs that you get from AlphaFold is not just, you don't just get the structure, you get a lot of extra information, because when you train a deep learning model, you train it with a certain task in mind. You optimize the parameters inside your neural networks with a certain task in mind, and that task is the optimization, for instance, the minimization of a certain cost function. And this cost function, or a loss function, as I have put it here in this slide, it can be the basic thing that we want to train for, which is a structure that very nice maps a structure ground truth. But there's also a couple of very interesting other extra costs introduced in AlphaFold. For instance, there are no physical constraints in the neural network itself about how it can fold or cannot fold, but there is a structural violation loss put towards the end of the network so that it can optimize to not have any structural violations implicitly. It is not put on there explicitly, but it is trained by optimization. And 2 extra things here at the bottom are that the model is required to give a confidence for each of the positions of its prediction. So this trains AlphaFold to be aware of what parts of the structure it is actually quite confident of, and what other parts it is not confident of. And especially in for end users, this is very important to be able to use the structures and to be able to draw certain conclusions out of that. So here you have an example of where, like, especially the inner parts of the protein are very high confidence here in blue, and then the parts that are a bit flapping outside of the protein are seen as less confident. And to the end user, this is very useful information. It also gives more. So this is more like the local confidence. So how confident am I that my amino acids are the correct distance of the other amino acids in the proximity. And this is more on a global scale. So what you could see here, what you could draw from as conclusion from this visualization is that there's a certain part that folds very well, here another part, and here another part. But AlphaFold has no idea how to put those three parts relative to each other. So the parts themselves fold up nicely, but how to put one part next to the other, it does not know. And this is very important to know in order to be able to use it. And so AlphaFold has been made available by Google. The codes to use it to make predictions and the weights for 5 models have been released. And since last week or 2 weeks ago, they even put CC BY 4.0 license on this, so it's available even for commercial use. Then let's talk about how to use it with Python coding. They made it available also on a Google Colab. So on Google Colab, you get resources allocated, including a GPU. These are like 12 or 16 gigabyte GPUs, which you can use for a limited time. And they have their codes all up and running. You can just put in a sequence there, run, and you will get your predictions. So this is very nice, but it's quite slow. And especially if you want to run a lot of sequences through it, it can be quite cumbersome. There's a bit a more user-friendly version, also faster and some more fine-tuning, which is called AlphaFold. This is by the Ovchinnikov lab, and they also run on Google Colab. And then there is the AlphaFold protein structure database, which has 100,000, right, a couple of hundred thousands of structure predictions for the proteins of all human proteins that we know, also 20 different other species. And I think lately it has even been, the numbers in the database have even been increased. So that's a very good resource. But often you still want to have your own proteins run through it. To give you an idea of how fast it has gone, that's since AlphaFold won the CASP14 competition at the end of November 2020. There has been quite a lot of stuff going on. So the first half year, I guess they have worked a lot on the release of the packages. In June, there was the release of the RosettaFold paper, and they also released their models. RosettaFold is a product of actually the group who is always ranked second now in the CASP competition. It's David Baker Lab, and they have their own prediction models, and they use the ideas from AlphaFold 2.0 to fine-tune their own approach, which is now called RosettaFold, and it's also still been used quite a lot. Also in parallel to AlphaFold, if AlphaFold gets a prediction wrong, perhaps RosettaFold can be used. It's a bit situation dependent. As a ground rule, AlphaFold is often better, but not always. So that was released in June and July. The AlphaFold publication came in July. The ColabFold was released a couple of days after. AlphaFold released their official Google Colab. The database was released at the end of July. The ColabFold paper was released in August. And then to give you an idea, at our university, AlphaFold was available at the end of August on our high-performance computing clusters. Then there is AlphaFold Multimer, which is a 2.1 version of AlphaFold, about which I'm going to talk later. That was released in October, and support of that was quickly added via EasyBuild, of course, later on as well. And then there is OpenFold that was released in November, which I'm also going to talk about more. But first, I'm going to talk about AlphaFold on the HPC itself. A bit more technical here. So the resources that are used by AlphaFold are actually not too limited. It's quite a heavy piece of software because it uses deep learning and it uses big databases for the homology searches. So the first step that I talked about was that multiple sequence alignment. So the sequence database search is very CPU memory intensive, and it actually wants a very high disk reading speed because it is looking up in data sets of over 2 terabytes of storage currently. And that's actually a step that takes quite a long time as well. And then in the second step, you have the deep learning models, which are very GPU memory intensive. So you need quite a good rig to run AlphaFold at desirable speeds. And so I'm going to talk a bit here about the memory limits and the time that it takes to do AlphaFold runs. So as a test, I selected 20 random proteins and ran them through AlphaFold, proteins with different sequence lengths, to see what influence that has on the memory consumption and the time elapsed. And did this on 2 clusters, one with V100s and one with A100s. And as you can see, the memory required is not completely in line with the sequence length. It's more dependent of how deep and how many similar sequences it finds, rather than just the length of the initial sequence itself. But you can see that often close to 64 gigabytes of RAM is required. I had one case where even a lot more was required. And if it's not available, then it just crashes. And then talking about GPU limits, when run on Google Colab on the 12 or the 16 gigabyte GPUs that are allocated there, you can run somewhere between 1,000 and 1,400 sequence lengths at most. And when you have V100s of 32 gigabytes, you can go almost until 3,000 amino acids. Now, I must say that the most interesting proteins are all in the ballpark between 0 and 1,000, and even more about the 400 or 500 length mark than anything else. But okay, there are cases where you want to have larger proteins run through it as well. And then you might run into memory limits. Looking at the time that it takes to do an AlphaFold run on those systems, I took the 3 different stages. So the structure database search and the sequence database search is here depicted in green. The prediction using the deep learning models is depicted in purple, and then the relaxation, so the fine-tuning at the end of the structures, is in orange. As you can see, the multiple sequence alignments initially takes the longest time for shorter proteins, but it does not explode when the proteins get too long, which is especially the case for the relaxation. And the prediction also increases quadratically. So when we're looking at short sequences, most of them run in less than an hour. So I have some extra timing information later on. And when you run it on the HPC, personally, I run it in an adapted script, which is a bit more user-friendly, so I can immediately submit multiple protein sequences at once. I just press enter, and all my proteins are aligned in the queue. I just wait a bit. As I said, most of the interesting proteins will take about an hour or even less to run through it. And then you get a lot of outputs. Depending on the script that you run, you can visualize some extra stuff. So what you will always get is the 5 structures that come out of each of the 5 models. You will get, you can visualize the multiple sequence alignments, so how many sequences you have and where they overlap with the query sequence. You can have the confidence nicely plotted out, as you have in the 2 other plots here. And then, okay, this was a timing that I was talking about earlier. I wanted to give you a comparison between the time it takes to run it on the Google Colab and what it takes on a specialized HPC machine. And you can see it's really a big increase in performance. Here I took the SARS-CoV-2 spike RBD as an example. And we're really talking about a multi-fold increase in timing. So that's a bit what AlphaFold is about. And that is what you're getting requests about if you have seen them pass by. So that is why. And so I can tell you it's not over here. You're going to get a lot more installation requests because people are doing a lot of stuff with this as a starting point. One of those is a protein complex prediction. So now, until now, I've been talking about individual proteins and how they fold. But protein complexes are also very much of interest. How do different proteins interact with each other? Especially in drug design, this is very important. And some people were playing around with the initial AlphaFolds, the 2.0, and they found that, okay, if we have a linker without a structure in between two parts of the sequence, AlphaFold will actually fold them up as two individual proteins, and they will interact with each other in some kind of way. There were some other people that found that you could actually just, instead of putting the linker there, programmatically separate two different chains, as they are called. And in October, Google DeepMind released AlphaFold Multimer, which supports this kind of separate chain structure prediction, but it is actually specialized to do so as well. And while the results are not always ideal, it's the first step towards more specialized systems here. Another thing that I showed earlier, this was released in November, is OpenFold. And this is PyTorch codes and also training codes for AlphaFold. So what DeepMind released was just the code required for inference, so for prediction itself. But if you want to train your model and specialize a model for, as in this case, multimer prediction, you need to have the training code as well. And that is what the lab of Mohammed Al-Qurashi has achieved very recently. So this is gonna probably have a lot of downstream applications with very specialized prediction models further on. There's a lot of research being done about single sequence prediction that do not have the coevolution restraint. And then one of the most exciting ones is even molecular dynamics, because we're talking now about protein structures in the lowest energy form in which they occur. But proteins are very living things and they change, and depending on interactions with other proteins, they will have a different conformation. And this is called molecular dynamics. And DeepMind themselves, they have already mentioned the molecular dynamics to be one of their next topics of research in which they're very interesting, because this might solve a whole new range of problems as well. So to conclude, there's a lot more coming. The impacts on healthcare and on drug design are going to be quite well, big. Most people think so. So that's why it's a very nice piece of software to have, and it's very nice to have a very nice, convenient way of installing it as well. So I hope you have an idea of what you've been installing currently, and if there is any questions about it, I am very happy to answer them.
Thank you to Jasper for that question. Sorry for the talk. I see Marcus has a question. I'll ask him to unmute and ask it.
Hi, Jasper. As you can think, we already have questions, people asking for AlphaFold. But we are a little bit concerned about the database, because it's fairly big and there are also fairly big files inside. Our first intention was to use CVMFS to distribute to the compute node, but because there are fairly large files in there, that is not the right way. So we are still thinking on which file system we should install the database. Do you know the I/O pattern which is done on the database? It's more like a streaming, or it's a random access?
I actually do not know this. Unfortunately, also installation-wise, it's Kenneth that has done it for us.
Okay.
So I was hoping he could give a bit more background information about certain decisions that he made, but I don't think he's present at current time. But about the access itself, I do not yet have a good view on that.
Okay, perfect. Thanks.
But it's a very, very good question.
Yes, it is.
Pass across to our next question.
You're not even going to try to pronounce my name? Thank you, Simon. This is purely a field or biology question. Timing is everything. I mean, AlphaFold arrived just as we were starting to look at, COVID and whatnot. So it's pretty obvious that the proteins from SARS-CoV-2 would be something they looked at. I think 2 of them were in the Cas14 set. But I was wondering if you knew if anyone had used AlphaFold to look at the different isoforms of the prion protein, or like why we get different aggregations and why these aggregations give different disease patterns for the different conformations of PRPSC.
I do not have the best view of this. I know that in our lab, we're also producing looking into drugs against targeting the spike protein, SARS-CoV-2. And what I've been asked to do already a couple of times is to have those variants, the Omicron, for instance, lately, run through AlphaFold to already have some hypothesis of why certain things are different now than they were before, and whether our drugs target them still in the correct way. So it's definitely very usable here, but I do not have a good view of how the rest of the world is using it so far, because there is so much going on with AlphaFold itself. It's hard to keep track of everything.
Yeah, I've been trying to find somebody who's worked on the prion protein with AlphaFold, but I haven't actually been able to find much of anything, which does kind of surprise me considering the nature of PRPSC and the way that the different isoforms or conformations clearly give different diseases and different disease patterns. It would have been a very ripe candidate for trying to understand why something happens.
Perhaps it has been done, but yeah, I do not have the biological knowledge to really make a hypothesis of whether it would work or not with AlphaFold.
Oh, it's short at least. I think it's like, what, 208 long or something, so it shouldn't be that bad to work on. Anyway, thank you. It was a brilliant talk. Very, very interesting. Thank you so much.
Thank you.
Pass across to Jorg now.
Yeah, thanks again for a brilliant talk. I really enjoyed it. Coming back to the database and file system question, because we installed AlphaFold recently on our new cluster, so that is still a little bit of an test bed, and we were using Ceph. I found out fairly quickly that the Ceph was limited to 1 terabyte. We have increased that. So now the 1.5 terabyte file is actually 1.5 terabyte. That said, if I'm doing a short read or whatever it is called, it's working perfectly okay. If I remove the option and I'm actually doing the whole thing, I still get a segfold. So when I was hearing about file size and limitations here, it's just a piece of information I wanted to share. So other people are not falling into the same problem and just download the database with the automatic script and simply say it is all working. Check it manually, because otherwise you might run into problems. And I think not all of the files actually have something like a checksum. So even if you unpack it and it doesn't really crash, it doesn't necessarily mean it has unpacked correctly. Thanks.
Okay. Are there any other questions?
I'm not currently seeing any more questions. So thank you for your talk, Jasper. Very interesting to learn more about AlphaFold. I know I'm one of the people who has installed it at our site, and interesting to actually hear what people might be doing with it rather than just me pushing this software out and going, nothing happens now. I just don't hear anything else about it. So thank you for your talk, and very interesting. Our next talk will be in about 20 minutes time.